6 million cells per cubic millimeter of blood. In females, it is little less, and the diameter is around 7.2 micrometers, average thickness around 2 microns, and the average volume is around 78 to 94 micrometer cube. And hematocrit, that is packed cell volume, in males is around 47%, and in female it is little less, that is around 42%. Lifespan of RBC is around 120 days, and the site of production of RBC is bone marrow, and the site of destruction is usually the tissue macrophage system. Since RBC has a biconcave shape, the advantage of these biconcave shape is that they can squeeze through the capillaries very easily, can withhold endosmosis for longer, and the larger surface area is provided because of the biconcave shape, which helps in quick exchange of gases across the capillary membrane. Some of the important functions of RBCs are: they're important for the transport of gases like oxygen, carbon dioxide. They're important for the acid-base balance because of the hemoglobin, which has amino acids in them. It is important for the formation of the bilirubin. The RBCs are responsible for the major portion of the viscosity of the blood, which is help into maintenance of the blood pressure, and it helps in the identifying the blood groups as it contains antigen on its surface. Red cell fragility. Red cells shrink in solutions with an osmotic pressure more than normal plasma. In solution with lower osmotic pressure, they swell, becoming spherical and lose their hemoglobin. So that process is called as hemolysis. The tendency to hemolyze is called as fragility. In hereditary spherocytosis, that is congenital hemolytic icterus, cells hemolyze more readily than normal cells in the hypotonic sodium chloride solution. The fragility is also high in G6PD deficiency. The red cells can also be lysed by drugs and infections. Hereditary spherocytosis. It's a common cause for hereditary hemolytic anemia. The membrane of RBC is made up of spectrin, and it is anchored to transmembrane protein by band protein 3 by protein called as anchorin. The hereditary spherocytosis is caused by the defect in band 3 spectrin or anchorin proteins, where their genes are mutated, and they'll be able to produce less amount of spectrin or anchorin or band 3 proteins. RBC indices. The number, shape, volume, and color of the RBCs indicate the quality of blood. Blood indices have got a diagnostic value in determining type of anemia. These blood cell indices are mean corpuscular volume. Mean corpuscular volume is the volume of single RBC in cubic microns. It can be written as mean corpuscular volume is equal to packed cell volume per 100 ml of blood divided by RBC count, which is in millions per cubic mm of blood, into 10, which gives the mean corpuscular volume in cubic microns. The normal value is around 87 cubic microns. Mean corpuscular hemoglobin. It's an average amount of hemoglobin in a single RBC, which is expressed as a picogram. So MCH, that is mean corpuscular hemoglobin, can be given as the hemoglobin in gram percent divided by RBC count in millions per cubic millimeter of blood into 10 gives the mean corpuscular hemoglobin into picograms. Normal value is around 29 picograms. The mean corpuscular hemoglobin concentration, that is MCHC, is nothing but it is the hemoglobin concentration in a single RBC, or it is the amount of HB expressed as percentage of the volume of RBC, which is eventually multiplied by 100. So MCHC can be given as the hemoglobin in gram percentage divided by the packed cell volume per 100 ml of body into 100 gives the MCHC values in percentage. Normally it is around 34%. Then color index denotes the ratio of hemoglobin to RBC. So normal value is around 1.